Well, myself and Andy, who Jim mentioned, have been interested in regenerative medicine for quite some while, and we were interested in regeneration of the human senses. And I guess it was probably maybe about 2.5 years ago now. Andy had found a paper in the literature which was referring to the loss of the sense of smell, and we started digging into it a lot more to it, because we'd never really come across the concept of losing the sense of smell before. We were familiar with, obviously people losing hearing and sight and that kind of thing, but we started digging around and thinking, well, is this something for which there is do a lot of people have to live with this? Is this something that affects a lot of people? And what's more broadly speaking the effects it has on them? And very quickly, because the first thing you have to do is, you can't just read the literature, you've got to reach out to the key opinion leaders in the field. And very quickly we came across Jim and his pioneering work in regenerative medicine for olfaction, and then also Bradley Goldstein, who we're working with as well. And again, the work that he's doing in this area as well. And through talking to them and others, people like Duncan at Fifth Sense, Carl Philpott, and a huge slew of other individuals, it became clear to us very quickly that this was a big problem. It affected a lot of people. It profoundly impacted people as well. And with every passing day, and when I speak to more people about this, it becomes ever more poignant how much this condition affects people. And it became clear to us through talking to Jim and Brad and others that potentially there was a way that we could actually look to tackle this. There was some underlying biology that we felt was viable to approach from a drug discovery perspective, and so we basically started working together to enable us to do that. And yeah, I mean, it's hard to believe, but it's so around 2 years, certainly a lot more. With COVID, it's a bit difficult to kind of gauge time sometimes.
Sometime in the past.
Yeah, sometime in the past.
Fantastic. Thank you. Listen, we'd love to get into the detail. And Jim, I understand you've got a presentation you're going to share.
Yeah, a few slides I thought that I'd share with everybody. Fantastic. Just to make sure they have a good sense of what we're trying to do and how we're going about it. So I'm just going to share my screen.
So for everyone, do pop your questions in. We're going to have a time for a specific Q&A. As Jim said, he's only going to share a few slides so we can have as much time as possible to hear your thoughts, to get your questions as best as we can. Great. Jim, go for it.
Great. Okay. So just to expand a little bit on what Tom said about the sense of smell in the olfactory system, what you have here is a cartoon illustrating what the human nose looks like. And the region of the sensory organ for the sense of smell is right here, right underneath the skull, right underneath this structure in blue, which is a part of the central nervous system, part of the brain that receives signals from the nose. And if you look with a microscope, and what we've cartooned here is all the different cell types that you find in that tissue. The one that's really important for the perception of smell are these neurons that have a long dendrite that go up to the surface. This would be the air cavity. So the sense of smell is based on chemicals wafting in on the air and interacting with these neurons, firing off a potential in the neuron that is then conveyed up to the brain via these axons. Those are what are responsible for the perception of smell. What's responsible for maintaining those neurons and the sense of smell are populations of what are called stem cells and progenitor cells that are found down here at the base of the epithelium. And there are two varieties. The ones in yellow are the ones that are actually active pretty much every day. And they are giving birth to new neurons, which will mature and form connections. And the reason for this is, unfortunately, that the system has to be in contact with the environment, which puts it at risk. It's vulnerable for being damaged. And so you need that capacity that's embedded in these yellow cells in order to replace the neurons that have died as a consequence of the things that they've been exposed to. But there's also a set of cells in blue. These are called horizontal basal cells, or HBCs, which are a kind of stem cell. It's a stem. When we talk about stem cells, we talk about stem cells that are present in a tissue and they're responsible for maintaining that tissue, so maintaining the olfactory epithelium. And then there are others that you've also heard about that are what are called pluripotent stem cells. These are the ones that everybody's very excited about because they can be turned into any of the tissues in the body. That's not what we're talking about here. We're talking about stem cells that remain resident in the tissue, but these are most of the time, they're actually dormant. They are sitting, waiting to be called upon. And what happens is that disease occurs when these do not activate properly under certain circumstances. And I'm going to show you a picture now of a slide now that will sort of indicate the extent of the problem. So it's not uncommon, it can be permanent. These are the kinds of things some of you may have experienced that can be associated with the loss of the sense of smell. And you all well know the problems that can occur, problems with nutrition, safety, quality of life, and perhaps even general health. And the problem is significant, particularly as we age. Roughly 50% of us between the ages of 65 and 80, and 75% of us greater than that age will have olfactory dysfunction. And this is shown here. This is age groups, and this is a score on the smell identification test, which is probably the best characterized of the sensory tests for seeing how well you can smell. So anyway, what happens is that when you're born, this is what your olfactory epithelium looks like. Each one of these very, very small brown dots, which you can't really see as individuals, is one of those neurons that helps you perceive when there are odorants in the environment. That's nice. That's what it should look like. Unfortunately, as we age, what happens is it starts to really get pretty moth-eaten. I've got a couple blankets at home that look an awful lot like that. And all of the, there's still some neurons around, but the numbers have been so badly reduced as a consequence of the vicissitudes of life that you can imagine here at someone 86 years old, it's not going to be working very well. And so one of the things that we've found is that there are actually two kinds of changes that occur. In one type, what happens is all of the neurons have disappeared. This is what, in red, those are the neurons, and that's what it should look like. Nice and pretty, a healthy population of these neurons. A healthy population of those yellow cells that I showed in the cartoon that are responsible for making new neurons. But in many of the areas that I showed you where there weren't any neurons left, what you see is that the epithelium has become degenerate. It's lost its neurons, and that's a problem. But it still retains, here in red, those horizontal basal cells, the reserve stem cells. And so if we could figure out a way to activate them, to kickstart them, then we might be able to restore the periphery of the nose to where it's more like what it was like when you were born. Never going to get all the way back there. Nothing is ever that easy, but that's the sort of the strategy. Now, so what we're trying to do then is that these horizontal basal cells, these reserve stem cells, these dormant stem cells, they make a particular transcription of protein in the cell that's responsible for controlling the behavior of the cell. And this is kind of a unique situation that only one protein in the cell is required to essentially serve as a switch to turn these guys on. If you think about something like cancer, there can be just tons of genes that have become aberrant in a cancer. Here, all we need to do is just activate the one or modify the one in order to get the restoration of these active stem cells and the olfactory neurons. And I'm going to show you a little bit of data from an animal model that shows that this can work. And so what happens is we can do a trick in animals where we cut out that gene that makes that protein and eliminate it from the horizontal basal cells. And this is if that gene remains intact, the red horizontal basal cells stay as horizontal basal cells. But if we clip out that gene, specifically in those horizontal basal cells, then you can see we've remade neurons and we've remade these active stem cells. And so this is essentially the basis for our strategy, is to figure out a way to get rid of this protein P63 selectively in the horizontal basal cells so that we can restore the population of neurons that's responsible for the sense of smell. And this is one of the approaches that we're using right now. You know, there are a couple models that you can use to try to modify the behavior of a protein in a cell. You know, there are conventional drug development stuff. Graham can talk about this in magnificent detail about how to go about developing a drug to target this kind of protein. The alternative approach is to interfere with the making of this protein in the cell and therefore eliminate it. And you use a kind of technique where it's a form of ribonucleic acid that's what we call an interfering ribonucleic acid, or RNAi. And what that does is that it will eliminate the ability of the cell to make that protein. So eventually it will go away and the cells will activate. And so this is an example in an animal model where if we target that protein with an interfering RNA, what we find is all of a sudden now those horizontal cells have become active and they're making other cell types within the epithelium. Whereas if you use an RNA that isn't targeting that protein, nothing happens. They just sit there. And so that's the kind of immediate strategy that we're trying to pursue. And so just to summarize really quickly, with normal aging and with other forms of olfactory dysfunction, you can get this stem cell exhaustion and depletion of the neuronal population. You still have the HBCs. So loss of P63 could be sufficient and certainly necessary and sufficient to activate the HBCs so that they get back in the game and they start making neurons and help restore the sense of smell. And so what we're doing is we're using a variety of in vitro models and animal models in order to understand this better so that we can, this is part of the usual drug development pathway. You have to try things out in an animal model and then in like in tissue culture, putting human cells in tissue culture and making sure that the thing that you're doing is not toxic and actually works before we can actually bring a drug to humans and try and see whether it's effective. And right now, this RNAi, it seems to be have a real potential that we're trying to exploit in order to develop a therapeutic for smell disorders. Graham, you want to pick up?
Thank you very much. And then Graham, I guess this is where you pick it up.
Yep. I'll pick up things from here. I don't actually have any slides.
No, that's fine.
I thought I'd just kind of maybe chat for a few minutes about things, if that's okay with everybody. Yeah. And I say it's maybe sort of 4 or 5 minutes, and then obviously we can move to some questions. So I've been interested, like I said, in this whole sort of regenerative medicine space for quite a few years now, and more recently on the human senses. And specifically what this is, it's about the discovery and development of drugs that can be used as new treatments for what are frequently called sort of degenerative conditions. Now I've used the term regenerative medicine a few times, so I should probably explain what that is, at least from my perspective anyway. And this is a new field or relatively new field of medicine, which essentially is looking to capitalize on the natural repair mechanisms which exist inside us and how we can use them to help the body fix itself when we incur damage to tissues or organs. So really what I'm talking about here is how we can modulate these regenerative pathways with drug molecules to provide therapeutic benefit to patients. And this is an area of research which has been attracting sort of steadily more attention from the biotech and the biopharma industries in recent times. Now, what we do know is that, well, we've known this for a long while, is that certain parts of the body are pretty good at repairing themselves. And we did touch on this aspect earlier. I mean, just for example, when we get a cut, those wounds close by themselves. Blood is replenished when we bleed. The liver, the gastrointestinal systems, they repair themselves effectively. So obviously we're composed of many more organs than that, and there's many other organs in the body which don't seem to do this. And so an obvious question to ask is why is there a difference in this ability to sort of self-repair, if you like? And so one way you could look at this is to think, well, it's likely driven by necessity, and the differing behavior is due to the fact that some parts of the body actually really need to repair themselves because they have to interact directly with the external environment or things we ingest. And I'm sure everyone knows we frequently incur damage due to everything that life throws at us and which we throw at our bodies. And the nose is a great example of this. The olfactory epithelium, and this is the area of tissue in the nose which senses or which contains the odorant sensing cells, and it's the one that Tom showed earlier. This is directly exposed to all manner of nasty things on a daily basis, and so those repair mechanisms have got to be there. So along, as we touched on this earlier as well, along with Jim and others, what we're doing at the moment is establishing a new company, which is called Merodia, and our mission is focused on developing therapies for people who are living with olfactory loss, impairment, or dysfunction. And our approach is pretty simple. It's based around looking to restart the regenerative process in a damaged olfactory epithelium, and essentially to give the stem cells there a bit of a kickstart to enable the tissue to repair itself again, and by doing so to restore the sense of smell. Of course, what we're looking to do is to capitalize on the years of work which Jim and our other academic co-founder Brad Goldstein have been undertaking in their labs, studying the olfactory stem cells, and to translate that pioneering research into a new drug which will be effective in restoring the sense of smell, and of course will also be safe and convenient to use. So there's another part of this as well, I think, that's probably become more obvious to us the more we've interacted with the community and with patients and scientists and many individuals. And that the other part of this that we think is really important as well is to raise awareness of anosmia and other olfactory disorders as problems, and to really raise awareness of how this affects people and the really significant impact this has on their day-to-day lives. And certainly conversations I've had with people has sometimes been resulting in this erroneous view that it's a minor inconvenience, and we really need to change that, and it's got to be a priority to change that perception. Not only just the fact you've lost your sense of smell, but also to get people to understand what the longer term consequences are, and Jim outlined those, of course. So looking at this from the perspective of the biotech and the pharma industry, there's not really been a whole lot of attention paid up until this point, though that is now starting to change. And it's certainly starting to change for various other sensory degenerative conditions. And the example that we frequently give here is of hearing loss. There's quite a lot of activity in the regenerative space for hearing loss, and it's obviously going to be interesting to see how that picture evolves for olfactory loss and dysfunction as well. So obviously we're hugely optimistic about this and really excited about the potential, but I think it is also important as well here to just sound a bit of a word of caution, and to try and balance the aspiration and that optimism with the realities of drug discovery. So the fact is drug discovery is high risk, and it can take over 10 years and hundreds of millions of pounds to take a new drug through to approval. So unfortunately, it's not like making a new car, which is somewhat more straightforward. There's a lot of unknowns in this process. There's also a lot of knowns, but there's also a lot of unknowns. And what we do know, though, is, and it's been estimated, that of the compounds that get into human clinical trials, about 1 in 10 of those make it onto the market as a marketed drug on average. Of course, as well as being high risk, this is also high reward. And I'm not talking about fiscal high reward here. What I'm talking about is reward for patients. And these new drugs that are now coming out onto the market make massive differences to the lives of patients and their families. And that's incredibly important. I mean, last year, just looked at this today, 50 new drugs were approved in the US by the FDA. I'm certainly, and I'm sure everyone else will be as well, is looking forward to the day when at least one of that number is for an olfactory disorder. So, as I say, it's a high risk process. It takes a while. It can be quicker, and there's ways that you can sort of enhance the probability of success. A lot of it depends on how much information you've got at the start of the process, how much information you've got, the data you've got, and the amount of research that's been undertaken beforehand. And even then, there are still no guarantees, but the more you know going in, the more chance you have of success. And this is why companies who are engaged in commercial drug research, they spend enormous amounts of time talking to patients, talking to clinicians and basic scientists about the condition that they're interested in, so that they understand as much as possible about what they're trying to do, and to do that, and in doing that, to basically build really solid, well thought out operational plans. And as part of this as well, they also ensure they're working with the top people, the leaders in the field, to give their drug discovery projects the absolute best chance of success. So, as well as raising awareness of olfactory loss and its consequences in more general terms, one of the challenges that we are facing here, I suppose that a challenge and an opportunity, I suppose you could term it, is we're working in an area where at the moment there are no disease modifying drugs at the current time. So, to some extent, you could say we're ploughing our own furrow, as it were, here. As I indicated, though, we can look to other areas of sensory regenerative medicine for comparisons, because they're likely to be common lessons and common learnings here. And I pointed out sensory neural hearing loss, and I think that's a really important comparator.
Thank you. And it's probably, I imagine it's an equally complex area, that we've got so many different types of
Indeed.
Smell loss and smell disorder. I imagine that it's got some equivalents in there.
There is, and we're starting to see common learnings across the area. And one of the points that we frequently make is the auditory tissue is not one that's so prone to regenerate naturally. And it's obviously also quite challenging to access. Whereas, of course, the olfactory tissue, the olfactory epithelium, one, it naturally regenerates, as Jim's explained, but secondly, it's relatively facile to access directly. So that should provide, I think, significant confidence that actually delivering a therapeutic to that target tissue would be relatively, well, sorry, I shouldn't say relatively straightforward, sure. But relatively speaking.
And when we've spoken before, you said that sort of the aim is going to be something like a gel you're looking at.
Yeah, no, absolutely. I mean, we've spent a lot of time thinking about this, of course, and talking to experts in the field and clinicians. And our longer term goal, or I think the longer term goal, would be some kind of patient operated device like a spray or something like that, where you give a metered dose at certain times, 2 pumps in the morning, 2 pumps in the evening kind of thing. But ultimately, I think there's a lot of basic work that needs to be done before that to establish things like optimal dosing, optimal duration of dosing, that kind of thing. And the best way to achieve that specifically will be through direct application of a therapeutic to the target tissue. And as I said, for olfactory tissue, that's actually relatively straightforward. There are not many conditions where you can actually do that. So, when you couple that, if you like, relatively facile delivery with the natural regenerative potential and ease of access of the tissue, you think, well, this looks pretty exciting. I mean, we've got an enormous amount of knowledge now as well, of course, through people like Jim and Brad about the olfactory system. We've got an enhanced understanding of the different pathologies which are associated with the various forms of smell loss, and of course the underpinning biological pathways, including how the cells regenerate. And so I think we can feel really optimistic that therapies for many forms of olfactory loss will be available in the coming years. And as this is something I as I'm sure everyone does, it's on this call, we think about this every day. And but from my perspective as a scientist who's working in the field of commercial drug discovery, I there's one take-home message that I'd give people, and that would be, we've heard you, we understand how this affects you, and we're going to be working as hard as we can and making our best efforts to bring new therapies to people who are living with olfactory loss or olfactory dysfunction. It's our number one priority.
Graham, thank you. That means a lot. I'm sure we'll be getting comments in about that. Thank you very much. We have lots of questions. I guess what I'm sort of taking out of that from both of those together, that it's a natural regenerative process, that you found the mechanism in there that could kickstart it again in certain areas. You've got the ideas of how it would be applied. Lots of bases in there and still a long way to go. Quite a few questions in here, so if I get you to keep your answers fairly brief. I know that's difficult for some of the complexity of this one. We'll try and cover as many as we can. Jill is asking a question quite a few people have asked. Would these new drugs help anosmia caused by different reasons? So you focused in on age-related issues there, Jim, in your presentation. Does it cover other areas as well?
So we don't know for sure. It's just that the form of pathology that we see, where there's this sort of neurogenic exhaustion, is the one that we're best able to model. That being said, the other forms of disease, like post-viral, that kind of thing, may also be amenable to this kind of an approach, targeting P63 in combination with something else. There have been some studies done, again, in animals, not in humans, suggesting that when you use a toxin on the nose, that normally would result in the kind of scarring that you see after viral or other forms of direct toxic loss, that you can end up with neurons. But these factors that seem to be important are not characterized yet. So that's another example of the work that needs to be done. I mean, one of the advantages that we have now is that we've got some really much more powerful approaches that will allow us to address these things more directly. The one thing that unfortunately I think will not be amenable to this form of therapy is likely to be congenital loss. That's its own sort of unique issue, and may have more to do with connection into the brain. Trauma will also be more difficult than some of these forms of smell loss that are taking place solely in the periphery.
So specifically what you're looking at is in the periphery, inside the nasal cavity.
Inside the nose, yeah.
For the moment, yeah. And so for people with congenital, sometimes it'd be about missing olfactory bulbs, for example.
Correct.
I haven't heard viral anosmia described like that before. You talked as like an attack or damage. And I think it's still ongoing though, isn't it, as to what exactly do viruses do?
Yeah, it depends quite a bit. So one of the things that, there was a paper that was just published within the last couple of weeks looking at COVID. And it may be that the smell loss that you see with COVID is of two sorts. There's the sort where someone recovers very quickly. And what this paper showed was that there were abnormalities in cell function of the neurons. The neurons were still there, but they just weren't operating properly. But there are also more severe infections with COVID, the ones that may cause persistent and long-lasting smell loss, that are most likely due to direct damage to the neurons and the stem cells that are present in the nose. And under these circumstances, the epithelium, which was olfactory, can change into the kind of epithelium that you see in the lower non-sensory, non-smell part of the nose. This is the thing that some people have been able to turn around and make it neuronal again, with some ill-characterized biological types of compounds, but it's that's even earlier days for us.
Okay. Question from Sarah is, if it's true that parosmia is evidence of some repair of the support cells, would it be appropriate to use the RNAi or the other method to enhance the process of the repair to cells? So if it's sort of the repair's happening, is this appropriate?
So that's a very good question. We don't understand very much about parosmia in humans. We do know a bit more about what looks to be parosmia in animal models of the disease process that can occur, where you damage the epithelium in the nose, you damage the nose a lot. And you still, because these are young animals, they recover very well. But their sensory function is distorted in subtle ways, and which is a lot of what parosmia is like. And under these circumstances, what we see is that the connections between the nose and the brain have gotten kind of mixed up a bit. Normally, they're very, very precise, and then they become somewhat imprecise, and that may be what's responsible for the parosmia. That being said, one of the things that we suspect, again, from these animal models, is that where the scrambling is worse is for those neurons that don't seem to be being stimulated during the regeneration process. That's an association. We haven't been able to sort of prove that directly, but that's what we suspect. And so, it may be the case that folks with parosmia will get better with time as the degree of their exposure to the environment increases, and perhaps even with very specific training efforts. And so, I think that there's a lot that we don't know, but under these circumstances, if you have a reduced population of neurons, more neurons are probably going to be better. And so there certainly would be damage to the periphery, and we might be able to repair that, which might allow the parosmia to resolve better and more quickly.
Thank you. Yeah, that makes sense. So it says that, thank you. I'm not going to restate what you said. Lots of questions in here. Oh, there's some questions in there about saying, what's your view when you do come to a point of having a treatment of some sort? Is it you sort of, you take it for a few days and it's repaired and you're done, or is it looking like a constant therapy that people have to take?
I don't think it has to be constant. Again, judging from the animal model, when we activate the horizontal basal cells genetically by taking out that gene for the p63 protein, cut it out, we've eliminated it completely, then those cells turn into active stem cells. And in mice, those active stem cells are active for at least a quarter, probably a lot longer. I mean, we just couldn't wait any longer. A quarter of the life of the mouse. And that's with just one tickle at one point in time. And so we think that it would only require like a brief period of treatment in order to regenerate these active stem cells. The longer you go, maybe you would get more and more of them. We don't know anything about how well we're going to distribute the drug, how quickly and efficiently it's going to work, all those are questions that remain to be answered. But I'm really optimistic that once you get those guys back in the game, they can stay active for a good long time. And we're one of the things that Brad is particularly focused on, he couldn't be here today, is the question of how do you help these active stem cells along. There may be other things that we will use in combination eventually that would improve the overall success.
Thank you. Quite a few people asking as well about testing, and I'm pretty sure you'd have a few volunteers today if you had something in a bottle you stick in. What's your Graham? You talked about this being a very complex process. When at the point does it go to human trial in your experience?
Yeah, I mean, at the moment, I mean, we think there's going to be probably when we get things started, maybe 2 or 3 years before we actually get into human clinical trials, because we've got quite a bit of research that we need to do to address the kind of points that Jim was making about how frequently you dose something, how often you do so, and how much, that kind of thing. And then it takes, of course, as you'll be aware, a number of years to go through the various phases of human clinical trials, phase 1, phase 2, phase 3, that kind of thing. Yeah, we're hopeful that with all of the knowledge that we've got, and with the experts that we're working with, and of course we've built up a great network of clinicians as well to help us move this forward, then we're hopeful that this can move forward relatively quickly.
Thank you. A couple of questions about you. In the meantime, somebody asking about a zinc supplement, somebody else talking about smell training as well. Jim, do you have a sort of a view on what zinc people can do that can be useful in the meantime?
So I know zinc has been suggested. There are other things that have been suggested that might help. I think the thing with zinc supplements is it really is only if there's a deficiency, a pre-existing deficiency because of diet or something like that, that the zinc supplements are likely to be effective. And you do have to be careful with zinc. Don't put it up your nose. It can actually damage the olfactory epithelium. So not so keen on zinc supplements. Smell training, like I say, there's evidence from the animal that would suggest that if you can get activity, you may help with connectivity. So activity may help with connectivity, and that may help restore the sense of smell. That may be a longer term kind of thing that you need to do it consistently for, I don't know, months perhaps. I just don't know. But I think it's a not unreasonable thing to approach. There are one of our colleagues who we have a good relationship with is a man named Thomas Hummel, who's in Dresden, and he's been a pioneer in the development and use of smell training, and has some data suggesting that there are that it's helpful. So it certainly couldn't hurt.
Thank you. Let's have a look. We're asking up to the last couple of minutes. I think it'd be good to finish on time tonight. I'm just seeing if there's anything else in here. Quite a few. There's a little chat going in the background about smell training, and Dan's just put a link there to resources on the Fifth Sense website for how you can do that for yourself. We'll have talks from there from Frauke, I think, is on the call as well, and videos to take you through it. That's not something you tried yet. Wow, lots of other bits and pieces. I'm sorry if I haven't got to everybody. Lots and lots of thoughts in there. Graham and Jim, thank you both very much for your time here tonight. Any closing thoughts from Graham? I think you first. Anything that's...
Thank you. Well, firstly, it's been a real pleasure to participate in this call, and thank you everyone for your questions, and I hope it's been useful and informative. I think, I'd just reiterate the point that I made, sort of the last point I made when I was speaking, as it were, which was, we really are, laser focused on this development of new drugs for people who are living with olfactory loss, and we really want to get that awareness up in the broader community and the like. I mean, I'd like to see a day, this is my personal opinion here, I would like to see a day where, as well as walking into an optician's to get a sight or a hearing test, you could get a smell test as well, and you could quantify your sense of smell. It would be so important in knowing when you were having maybe some decline or some other issues. And this will, all of this is going to come together along with the development of new therapeutics. There's probably loads of people out there who are living with a mildly hyposmic or approaching anosmic, who perhaps maybe aren't so aware of it, and we want to be able to help everybody.
Thank you. And Jim, we've had a request asking about your paper that you mentioned.
Which one?
Somebody just said, Jim, could you mention the authors of the paper you mentioned? Is it published yet?
So my memory for names is not what it used to be. The folks, I'll tell you what, I will send to Dan the papers that I've referenced, so that's fine. So
fantastic.
If I can just have one last word, or
please do.
Last words. We are, in a sense, very lucky here. Because the biology is so much simpler for us than it is in so many other of these tissues. The fact that we can target a single protein, a single gene, and have an effect is almost unheard of in biology. When somebody has cancer and they give them Taylor's disease or multiple myeloma, they commonly give a drug called bortezomib. Well, that's just like a sledgehammer, right? That it affects a process in the cell that is so important for all aspects of the biology of the cell. This is focused. This is like a laser advantage that we have to go after this one target. And so that's one of the reasons why I'm particularly excited and optimistic about it, because in my simple-minded way, I can see a path forward that's not as incredibly difficult and as incredibly complicated as a question like cancer is.
Thank you very much. We're going to come to Duncan Burke, the Fifth Sense founder, in just a second, but just to say that we will send around an email after the talk today. We will go and have a look through the chat box as well. If there's any questions which are unanswered in there, we can point you towards some resources or talks that we have in place, then we will do. Duncan, go for it. You should be able to unmute yourself now that you can. Great.
Thanks, Tom. I'd just like to say, thinking back to 10 years ago, when I first started setting up Fifth Sense, to think that, I'd be sitting listening to you, Jim and Graham, talking about the work that you're doing, and the fact that we are on the path to getting drugs to treat this condition. That's something I could only have imagined back then. So it's incredibly encouraging and incredibly exciting, and thank you both very much indeed for coming along this evening and sharing your work, and we'll look forward to keeping in touch and keeping our members informed of developments. Thank you.
It's been a real pleasure, Duncan. Thank you.
Really a pleasure. Happy to have done it.